Welcome back to Intro to Physical Anthropology. I'm your instructor, David Leitner, and this week we are talking about genetics and the human diaspora. How are we all related? Let's get started. So, we can get genetic data from a couple of sources. We can look for most recent common ancestors by sort of looking at living populations and comparing similarities and differences, constructing a phylogenetic tree. We can also isolate DNA from fossil hominins and look for differences there. We can also do a combination of the two, and we're going to talk a little bit about that today. So, there are various kinds of DNA that we can use and various kinds of proteins that we can use as well. Proteins are another way that we can trace lineages back. Mitochondrial DNA is very important because, as you'll remember, mitochondrial DNA doesn't exist in the nucleus. It exists in the ribosomes, in the mitochondria. And there are many, many, many copies of it in a cell. So it's more plentiful than nuclear DNA. It also is only passed on from mothers to daughters. So men, you have your mother's mitochondrial DNA, but you won't pass it on. Women, you have your mother's mitochondrial DNA and will pass it on. It's clonally passed on. So it is the exact same copy. But men, you can only pass it on to your daughters. So, now, when we trace the various lineages of mitochondrial DNA back to their source, we come up with a so-called mitochondrial Eve, who lived probably between 120 and 160,000 years ago. Now, that's a little different from the 200,000 years that we've been talking about for fossil evidence, but there's a reason for that. That's not surprising. We are not literally looking at the very first human here. That is not what's going on. What we are saying is the founder of the lineage that includes every human alive today, in other words, the lineage that has survived for all this time and hasn't died out, lived around that time. There were lineages that existed before that, but which didn't continue on to the present. So the oldest lineage we can trace doesn't mean there was just one individual in the population. There were lots of individuals. They just weren't part of this lineage. This is the surviving lineage. In the same way, we can talk about other Eves, like a European Eve, or a British Eve, or Chinese Eve, although that starts getting a little dicier because we have to keep into account that we don't necessarily know where that person was, right? Where people are today is not where their ancestors were necessarily. This is one of the reasons why you need to take 23andMe and those sites with a grain of salt, because they are only measuring genetic variation in the populations in those countries today and comparing it to you. Your ancestor you share with people in that country may not have been from there at all. Anyways, so mitochondrial DNA gives us a great sort of look at lineages. Similarly, the Y chromosome for men, which only men can inherit Y chromosomes, so it always goes patrilineally from father to son, never to daughters. This gives us a similar sort of picture, but for a completely different molecule. It has similar advantages to mtDNA. Like I said, it is it's descended unilinearly, that is, from just one side of the family. In Y chromosome case, it's the male. mtDNA, it's the female. And it has some regions that are more highly variable than others, which makes it useful for clocking relatively short time spans like 200,000 years. When we do the analysis, we end up with at least 18 major haplotype groups. A haplotype is just that. It's a group of similar lineages, basically. And these haplotype groups converge on an ancestor probably around 236,000 years ago. Again, doesn't jibe exactly with 200,000 years, but again, we aren't necessarily looking at the first Homo sapiens, but rather the first individual in our past that had all the elements of these haplotypes, that these haplotypes are descended from. Okay. Same thing as with mtDNA. Interestingly, the non-African most recent common ancestor is very recent, much more recent, and jibes with a lot of our data on the expansion of Homo sapiens out of Africa. This just gives you an idea of what these lineages give us. I'm going to pay attention to the two mitochondrial lineages here, especially. On the left, you have the mitochondrial DNA comparisons. That is, all comparisons of modern, existing, like contemporary mtDNA samples. Okay, no ancient DNA here. And that gives us a picture where we've got a series of splits in the lineages, where you've got some that include African and non-African lineages, and the others that are African only, which we would expect because of founder effect, right? Those dates also sort of give us some tantalizing time frames too. 171,000 years, that's not so far off. And the ancient mtDNA gives us a pretty similar picture. Imagine, though, this is on a wider time scale, so, you know, the node C would be node A here in the other, in the left-hand chart. But in this right-hand chart, it's just one piece. And here we're seeing the shared mtDNA of, they're calling them Western and Eastern Neanderthals. The Eastern might be Denisovans. I'm not sure in this case, actually. But regardless, we all share a common ancestor about 600,000 years ago. That jibes more or less with the fossil evidence as well. Okay, since we're talking about DNA, the last thing I want to talk about is introgression. I've mentioned this a little bit more before, but introgression simply means that gene flow. That's all it means. It's genetic material from an archaic population that is then reintroduced into a newer population. In this case, it's always polymorphisms, okay? We use these polymorphisms to construct phylogenetic trees so that we can see where these crossover events must have occurred, because there'll be interruptions in this tree. As you can see here, so here we've got a representation of an ancestor group that it represented by the red dots. That's their genetic diversity, right? And then there's a split. And in one direction go Homo sapiens, and from that point on, novel Homo sapien polymorphisms are represented by gray, and novel Neanderthal Denisovan polymorphisms are represented in yellow. So you can see they go along their separate ways, and then there's another separation between Neanderthal and Denisovan. Neanderthals in green, Denisovan in blue. So what you've got here is, on the Denisovan line, you've got the DNA of its common ancestor with all of these hominins in red, the common ancestor with Neanderthals in yellow, and then its own distinctive derived trait polymorphisms right here in blue. What these arrows indicate are moments of introgression, so that is interbreeding. So you can see there can be multiple from the same species. They'll be they represent them in slightly different colors because they may be different polymorphisms. There isn't just one polymorphism that's different in each species. There are many, many, many. And that's what introgression is. That is how all that Neanderthal and Denisovan DNA has gotten into our genomes, those of us outside of Africa, with ancestors outside of Africa. That's it for this lesson. As usual, take care of yourself, have a great week, and I will see you soon. Bye.